Now, you may want to put a title at the top of your notes that says the cell cycle because this is kind of in the middle of a PowerPoint. It is a continuation from yesterday. Thank you again for being patient with the projector situation. I just feel like this is the best way to do this without me standing up front. This way, I think, is also beneficial to you because you can take breaks if necessary or pause, etc. So, definitely during this, if you do not understand something, ask questions. So, let's start with the fact that the mitotic phase of the cell cycle is alternating with the longer interphase. In your lab yesterday, you found out that interphase was the majority of the time that was spent in the cell cycle. Uh, but let's get a little bit more specific today and look at let's get a little bit more detailed today and look actually at what happens in each part of mitosis. So the M phase or mitosis consists of mitosis and cytokinesis. Interphase consists of cell growth, copying of chromosomes, basic preparation of the cell for cell division. And I want you to notice on this image again, interphase is all of this, and mitosis is just this small little piece of the pie. Here it is in a larger view. Now, interphase is about 90% of the cell cycle, and it can be divided into subphases. First, you have first gap, G1. You have synthesis, and G2. So the cell grows during all three of the phases, but the only part of that when chromosomes duplicate is during the S phase. And then the cell divides, and that's the M phase. So in interphase, you have the growth, the replication of chromosomes. When we say growth, we mean that new organelles are formed, new enzymes, new membranes, because if you think about it, if we are duplicating a cell, we need double the amount of everything we started with. And then mitosis has two parts to it. In the mitosis or mitotic phase of mitosis, the cell will separate and divide the chromosomes. And sometimes that is called nuclear division. And then in the second part of it, cytokinesis, there's actually a division of the cytoplasm in the organelles. And then the daughter cells may repeat the cycle. So let's look at each phase a little bit closer. And let's start at G2 of interphase because that precedes mitosis. So you can note in the picture, and it will be very important for you to look at the pictures. You also have images in your textbook that you can review. The nucleus is very well defined, and you'll see that the nuclear envelope is present, and you can see that the pores are in that image on the right. There are also one or more nuclei present, and you can see that in purple right here. There are two pairs of centrosomes. Okay, the plant. Remember that plants lack those. Those are not present in plants. And then there are a bunch of microtubules surrounding those centrosomes, and we call those asters. The DNA has replicated at this point because G2 is after the S or synthesis phase. And the form in which the DNA is present is as chromatin material. During prophase, what will happen in the nucleus? And we will split this up into nucleus and cytoplasm. So make sure that you differentiate in your notes. The nucleoli will disappear and you will start to see chromosomes becoming visible. And each chromosome itself is made up with, or up of, excuse me, two sister chromatids that are joined at something called the centromere. And the way that, if you recall in interphase, the DNA was present as chromatin, and now it's present as chromosomes. I want you to see here how that change actually occurs. So this is a diagram showing the process of DNA organization or DNA folding. First, the DNA is organized in chromosomes. And what it does first is you